I'm going to be addressing susceptibility of children to COVID-19. I want to start with a comment that it's important to recognize that data continue to be preliminary and change almost daily, and for the most part, definitive conclusions cannot yet be drawn. Now, there has been press coverage implying that children cannot be infected by SARS-CoV-2. However, we can definitively state that children can become infected. This slide shows COVID-19 data in children in the United States as of November 11th, when there were 7.6 million cases. While the number of cases in children is less than in adults, over 700,000 children 0 to 17 years have had lab-confirmed infection, accounting cumulatively for 9.2% of all infections. Now, children are less likely to be tested than adults as they are less likely to be symptomatic, so the true prevalence in children is really not known. And there's been a striking increase in prevalence of infection in children over time. And this chart shows you the percent of COVID-19 cases that are in children, cumulatively shown in purple, and in the past week in green. In April, only 2.2% of infections were identified in children, but by September, this increased to 10% cumulatively and almost 16% weekly. The lowest rate of infection, 1.7%, is among younger children aged 0 to 4. And as we will discuss, there may be a difference in infection rate between younger and older children. And you'll note in this figure from the CDC, school-age children between age 5 and 17 are grouped together. This slide shows you trends in infection over time focused on almost 300,000 school-age children, now broken down into age 5 to 11 years, the green arrow, and age 12 to 17 years, the red arrow. And you can see both age groups show a substantial increase over time as mitigation restrictions eased in the summer. But the incidence among adolescents remains almost double that in younger children. You'll note that the highest rate of infection is among young adults who appear to be playing an increasing role in transmission. These are data looking at transmission dynamics by age group in hotspot COVID-19 counties in the US. Prior to the counties being labeled as hotspots, which is the vertical gray line, the highest percentage of positive cases using a 7-day moving average was in those 18 to 24 years in red, followed by age 0 to 17 years in brown. This increase in infection in children and youth were then followed several weeks later by an increase in infection in older adults, 45 to 64, purple, or 65 or older, green. Thus, infections among young people and children occurred prior to the wider community of older adults. Now, these are data from a meta-analysis of contact tracing studies by Viner and colleagues that was published in JAMA. And they looked at the odds of developing secondary infection after exposure to an infected index case, and they stratified this by children less than 10 in the red box and adolescents 10 to 19 in the blue box, compared to adults over 20. So looking at the odds of secondary infection in 8 studies in children less than 10, you can see there's a 52% reduced likelihood of secondary infection compared to adults, that red circle. However, in 5 studies in adolescents 10 to 19, there was not a significant difference in risk of infection between adolescents and adults in the blue circle. Now, while many studies suggest that children, particularly younger children, have lower susceptibility, I do want to note that other studies have reported similar secondary infection rates as in adults. These are data from a CDC household study in Tennessee and Wisconsin, where 101 index patients were identified less than 7 days after symptom onset, and 191 household members were tested. And as you can see in the red box, secondary infection rates were similar in children less than 12, 12 to 17, and older adults. And this is a second household transmission study In Wisconsin and Utah, where 58 index patients were identified less than 10 days after diagnosis, testing 88 household contacts, and the secondary infection rate was 28% in pediatric and 30% in adult contacts. So while a number of studies suggest lower susceptibility in young children, definitive data are not yet available. Now, there have been a number of hypotheses for why children may have lower susceptibility to SARS-CoV-2 infection and milder disease than adults. First, related to the receptor for the spike S protein on the virus, is potential age-related differences in cell surface viral receptor expression. Second is the hypothesis that children may have some immunologic advantages, including potential cross-reactive T-cell immunity due to common cold coronaviruses and the active thymus in young children. And finally, severe disease is often accompanied by a cytokine storm of pro-inflammatory cytokines, but children have an anti-inflammatory cytokine predominance. So we'll go into some detail on this. The first hypothesis for presumed lower susceptibility is potential differences in SARS-CoV-2 receptor levels by age as a biologic mechanism. And in order to do this, to understand this, we need to look at how infection occurs. Two cellular proteins on the cell surface are involved in viral entry: angiotensin converting enzyme 2, ACE2, and type 2 transmembrane serine protease, TMPRSS2. And this simplified cartoon illustrates infection. First, the spike protein on the virus binds to ACE2 on the target cell surface. The cell membrane TMPRSS2 protease binds to and cleaves both the ACE2 receptor and the spike protein, activating the virus for cell entry. And then finally, cleaved ACE2 and activated spike protein enable cell fusion. Now, two studies, and actually there's been a third, suggest that there are lower levels of ACE2 and TMPRSS2 expression in the respiratory tract of children. The graph in the middle is an evaluation of ACE2 gene expression from stored nasal epithelium from a study of asthma in persons 4 to 60 years. And you can see ACE2 gene expression was age dependent, being lowest in children aged less than 10 years, that red box. A second study using public gene expression datasets to look at differential expression of ACE2 and TMPRSS2 in nasal and bronchial airways have the figures on the left, which show that children in black had significantly lower expression compared to adults of ACE2 in the first figure and TMPRSS2 in the second figure in both the upper nasal airway and the lower bronchial airways. A number of other possible hypotheses for lower susceptibility in children have been proposed. It's been hypothesized that human coronaviruses associated with the frequent common colds we see in young children may offer some protection due to cross-reactive T-cell immunity between SARS-CoV-2 and other coronaviruses. And there was a recent paper in Science that evaluated blood samples from prior to 2019 that found memory CD4 cells that react to SARS-CoV-2 epitopes that cross-react with corresponding homologous sequences common to circulating common coronaviruses. In contrast, neutralizing antibodies are human coronavirus species specific and do not appear to show cross-reactivity against SARS-CoV-2. Another hypothesis is that the active thymus activity and T-cell function, especially regulatory T-cell function in children, may be associated with a reduction in infection, and if infected, infection severity. As shown in the figure on the left, in young children, T-cell development is functional and the T-cell pool diverse, while the thymus involutes with age with loss of diversity and function. Additionally, children have high levels of anti-inflammatory cytokines like IL-2 in blue and lower levels of pro-inflammatory cytokines like IL-6 in brown. And the increasing pro-inflammatory cytokine predominance with age has been called inflammaging. And this may also make children less likely to have severe disease due to lower baseline levels of inflammatory cytokines, reducing the likelihood of cytokine storm associated with COVID-19. Now, a related issue to susceptibility is transmissibility. Can children with SARS-CoV-2 infection transmit to others? And there are mixed data here. While children can definitively transmit, the extent of transmission is unclear. Now, the role that children and young people play in transmission of SARS-CoV-2 depends on multiple factors. First is the risk of exposure to an infected individual, and children have been less likely to be exposed during mitigation interventions. Second is the probability to become infected once exposed, and we just discussed some data that suggest young children may have lower susceptibility to infection. Third is the extent to which symptoms develop that may lead to testing, and children are more likely asymptomatic, hence less likely to be tested. Fourth is the propensity to make potentially infectious contact with others, that is the number of social contacts across age groups, and school reopenings may increase this possibility. And finally, transmissibility is associated with the extent to which viral load in an infected child becomes sufficiently high to transmit and the duration of infectiousness. Now, once infected with SARS-CoV-2, the data indicate that viral replication and viral load is similar in children and adults. And this is a study that evaluated viral load extrapolated from cycle threshold of the RT-PCR test. At the time of diagnosis in 53 children, 352 adults with symptomatic COVID-19 tested within 5 days of diagnosis. And there was no significant difference in viral load between adults and children when looking at age in a continuous fashion in the scatter plot on the left, and by age category 0 to 11, 12 to 19, 20 to 45, and over 45 on the right. But what about asymptomatic infection? Well, viral load does not appear to differ between asymptomatic or symptomatic children or younger versus older children. And these are data from a prospective study of 382 children and adolescents from 204 households with an infected index case. On testing of the child contacts, 293 or 77% were infected, with 30% of these infections being asymptomatic. And they did quantitative PCR on 178 samples. So viral load did not differ by age group 0 to 5, 6 to 13, or 14 to 20 on the left, or by symptom status in the middle box, 3.8 log if asymptomatic, 4.1 if symptomatic. And on the right, in those children with symptoms, the highest viral load were the 3 days before to 3 days after symptom onset, decreasing thereafter, and didn't differ by age group. 0 to 5 is in purple, 6 to 13 in yellow, 14 to 20 in green. Now, transmission from child to child and child to adult can occur, and these are data describing a SARS-CoV-2 outbreak in a nursery in Poland. So following notification that a staff member had been exposed, PCR testing was then conducted among infant, staff, and family members. And the circles here represent infected individual with the age inside of the circle. Red circles indicate individuals with symptoms, and the line indicates household members of the case. The exposure resulted in 29 confirmed infections, 27% of those tested. So there was an index case and a 52-year-old staff person who had become infected from a household member. Another staff became infected, resulting in infection in the staff's children. 8 infants in the nursery became infected, none with symptoms, and that led to infection in 12 adult and child family members of these infants. So this illustrates that transmission can occur among infants and children and staff in childcare settings and can extend into the household and community. And in case you didn't like that that was from Poland, a similar outbreak was reported in the MMWR in the United States. 12 children acquired SARS-CoV-2 in childcare facilities. Transmission was documented from these children to over a quarter of non-facility contacts, with one parent being hospitalized, and transmission occurred from both symptomatic and asymptomatic children. In summary, children are not immune to SARS-CoV-2. They are susceptible and can be infected. However, some but not all data suggest that younger children, particularly those under age 10, may be less susceptible to infection than older children. And this lower susceptibility, if real, may be mediated by lower levels of SARS-CoV-2 receptors in the upper and lower respiratory tract of young children. However, data are confounded by the lack of testing among children due to higher rates of asymptomatic infection, and also the effects of mitigation on the likelihood of exposure to children to infected individuals. And as mitigation measures decrease, there has been an increase in pediatric infections. And clearly, further research is needed to better define susceptibility and transmissibility among children. Thank you very much for your attention.